I'd like to start by thanking Olink for the invitation to record this talk for the Olink Proteomics World event. My name is Ferhan Qureshi, and I'm the Senior Vice President of Biomarker Research and Development at Octave Bioscience. The title of my presentation is From Biomarker Discovery to Clinical Translation, the Multiple Sclerosis Disease Activity Test Product Journey. As an introduction, neurodegenerative diseases, including multiple sclerosis, Parkinson's, Alzheimer's, and others, represent a vast emerging field of innovation. These are complex, chronic, high-cost diseases with devastating outcomes. They are generally poorly characterized, and there's a lack of metrics and tools to help manage treatment. Focusing specifically on MS and the burden of this devastating disease, it is estimated that there are close to 1 million patients with MS in the United States, with an average of $65,000 in costs per patient per year. This is largely driven by disease-modifying therapies. MS has a very complex therapeutic landscape. There are over 20 approved therapies with diverse mechanisms of action. Notably, safety is often inversely correlated with efficacy. Historically, there have been limited tools to optimize therapeutic management and select the right therapy for the right MS patient at the right time. This leads to lots of trial and error in patient care, and that can result in suboptimal outcomes. Octave is creating a new paradigm with objective measurement tools to better manage neurodegenerative diseases, starting with MS. The goal of the Octave Precision Care solution is to improve outcomes and address unmet needs across the entire MS ecosystem, for physicians, for health plans, for pharma, and most importantly, for the patients. Our solution consists of 3 dimensions, each of which delivers novel measurement tools to expand disease insights. The first dimension is biology and consists of a multi-analyte biomarker test, the MSDA test, which I will discuss in depth today. The second dimension is our MRI Insights program. This program utilizes AI and machine learning for the QC of raw images and advanced analysis. This is coupled with neuroradiologist review to contextualize the findings and deliver structured reports to the ordering clinician. The third dimension is our Clinical Insights program, which incorporates continuous monitoring using mobile applications to track both symptoms and therapy adherence, as well as passive monitoring using sensors and wearables. MS-certified nurses also routinely engage with the patients in between their neurologist visits. While each dimension, including our MSDA test, can stand on its own, the Octave platform allows for the integration of findings across all dimensions for individual patient insights and allows for population-level analysis as well. The current standard of care for MS relies primarily on MRI interpretation and clinical examination of the patient. These are both lagging indicators and consist of retrospective analysis of damage that has already occurred. That damage can accumulate over time, leading to irreversible disability. They are mostly qualitative and subjective, often with poor correlation to one another, which is referred to as the clinical radiographic paradox in MS care. At Octave, we believe biology is closest to the truth. Octave is developing novel blood-based biomarker tests using cutting-edge proteomic tools and techniques. Measuring proteins provides a more sensitive and dynamic assessment of underlying biology and mechanisms such as neuroinflammation and neurodegeneration. These biological processes lead to disease activity and disease progression in MS. Disease activity and disease progression are distinct but closely related. Disease activity waxes and wanes over time and is characterized by relapses and periods of remission. Disease progression is the accumulation of disability over time and can occur as a result of relapses as well as independent of relapses. Our intent is to optimize the care of patients to address unmet needs related to both disease activity and disease progression. These include selecting the right therapy, monitoring for response to the therapy, monitoring for relapses, as well as measuring smoldering disease that can lead to silent progression over time. The MSDA test, the Multiple Sclerosis Disease Activity test, has been validated and has been clinically launched. Our multiple sclerosis disease progression test development is well underway. Using proteomics, we can achieve a more sensitive assessment of multiple sclerosis disease activity and disease progression. This leads to earlier insights, which enables faster interventions and ultimately improved outcomes for the patient. This slide presents a summary of the multiple sclerosis disease activity test. It's a minimally invasive blood serum test, and it was developed from its inception specifically for multiple sclerosis. It has been both clinically and analytically validated, and it integrates the measurement of 18 protein biomarkers. Those biomarkers are used to report on 4 key biological pathways: immunomodulation, neuroinflammation, myelin biology, and neuroaxonal integrity. We provide a quantitative overall disease activity score that's scaled from 1 to 10 with intervals of 0.5, and there are thresholds that correspond to low, moderate, and high levels of disease activity that aid in interpretability. We're set up for routine clinical use in nearly 100 clinics across the United States, and the test is used to provide deeper insights for both clinical practice and for pharma. This diagram represents a 5-year process at Octave to conceptualize, develop, manufacture, validate, and launch the MSDA test. A key aspect of this process was access to deeply phenotyped MS samples that had been consistently processed and stored, along with obtaining associated annotated clinical, radiographic, and demographic information on each subject. Over a dozen deeply phenotyped cohorts were accessed in collaborations with academic centers. There were 4 stages in this process. In feasibility, we first refined our target product profiles to ensure the assay we were building would address unmet needs in the ecosystem. We identified candidate biomarkers and also evaluated multiple proteomic platforms. We performed our first proof of concept biomarker study in extreme phenotypes to evaluate biomarker signatures between multiple sclerosis patients and healthy controls. In our discovery and development phase, Olink was utilized heavily along with other immunoassay platforms to increase the total number of biomarkers we were screening to over 1,400. A series of studies were conducted to iteratively select the top performing 21 proteins for our custom assay panel relative to endpoints of multiple sclerosis disease activity. We also prepared for validation and launch. We received our CLIA license, our CAP accreditation, and we secured access to critical reagents. We manufactured 2 lots of the custom assay panel with Olink and performed additional studies to further refine our algorithms. The custom assay panel was then analytically and clinically validated. We then beta launched the test in a single clinic for approximately 6 months to refine the logistics from ordering, collecting samples, all the way through reporting prior to a broader clinical launch. We're now conducting an ongoing continuum of studies to further demonstrate generalizability, clinical utility, and to expand our body of evidence. There were several advantages to using the Olink platform for our program. The first was the extensive library. This allowed us to cast the net widely. Key content was available representing immune response, inflammation, neurology, and other relevant pathways. Also notable was the low volume requirements, which is essential when accessing precious banked samples from collaborators, as described on the previous slide. PEA dual recognition offered best-in-class specificity and was highly sensitive. This was essential for our application, including the measurement of proteins that are predominantly expressed in the CNS that we are measuring in the periphery. We were able to leverage service offerings by Olink to run large cohorts of samples with rapid turnaround time, and Olink provided excellent support to bring the methodology in-house so we could perform it in our lab. Olink had custom panel development formats that matched our specific application, including the ability to add novel targets to our panel. They offered optimization of the assays and then collaborated with us to execute a fit-for-purpose validation based on our specifications. Absolute quantitation was added for all of the analytes on our 21-plex. We also calibrated our gold panel to what was considered gold standard assays that are performed on other platforms. One example is neurofilament light and is presented in the plot on the right-hand side. This data was presented independently of Octave earlier this year at the ACTRIMS conference. In this study, investigators from UC Irvine observed very strong correlation between the Simoa NFL assay versus Octave's MSDA NFL assay as measured in clinical patient samples. To date, we've manufactured and bridged 4 lots of the custom assay panel with Olink, each time scaling up to larger manufacturing sizes. The MSDA test is performed as a laboratory-developed test, or an LDT, in Octave's CLIA-certified and CAP-accredited laboratory. These are the 21 biomarkers that were selected for the custom assay panel, 18 of which are utilized in our MSDA algorithm. For those of you familiar with neurology, multiple sclerosis, and CNS biomarkers, several of these could be considered the usual suspects: neurofilament light, or NFL, GFAP, GFAP, CXCL13, and BAF. In addition, several novel targets were identified in our series of studies. Establishing both correlation with disease activity endpoints and investigation of causation, or biological relevance, were both central to finalizing the selection of these biomarkers. Statistical significance of the biomarkers was established using our data science and machine learning pipeline. Both univariate and multivariate associations with relevant endpoints were established and replicated across multiple studies. This slide summarizes our systems biology approach to corroborate each of the selected proteins' biological relevance to multiple sclerosis. We incorporated computational biology models, automated curation of ontologies, protein-protein interaction modeling, gene set enrichment, and spatial expression profiling. We found that the features that were selected based on statistical associations with endpoints broadly represented the complexity and heterogeneity of the disease, as represented by the list of pathways and processes. Notably, several of the biomarkers were also associated with mechanisms of action of therapies currently in use and those that are in development. The work I've described on the past few slides, incorporating both correlation and causation to select the 21 proteins for our custom assay panel from a much larger pool of candidates, over 1,400, was published earlier this year in Nature Communications. Prior to manufacture and then executing the analytical validation, there was extensive work performed by Olink and Octave to optimize the performance of the individual analytes on the custom assay panel. This included dynamic range optimization, as well as adding absolute quantitation for each of the biomarkers. A dual lot and dual site approach was incorporated into this process to evaluate accuracy, precision, sensitivity, interference, specificity, robustness, among other parameters. Accuracy, sensitivity, and precision were all assessed using endogenous protein material as opposed to recombinant. Interference experiments included characterizing both routine sample conditions, such as hemolysis, lipemia, and bilirubin, as well as evaluating for interference of common drugs and MS therapeutics. I mentioned the complex therapeutic landscape with over 20 disease-modifying therapies available, representing diverse mechanisms of action and routes of administration. Stability experiments were performed to establish conditions for processing, storage, and transport that reflected real-world use in the clinic. Diurnal and ultradian variability was characterized to investigate changes in biomarkers over short periods of time that are independent of disease processes. Results from these experiments, in some cases, disqualified biomarkers for inclusion in our models. For example, growth hormone for high diurnal variability and COL4A1 for high levels of imprecision were both excluded from consideration for our models. Analytical validation was first performed for the individual proteins, and then upon completion of our clinical validation for our algorithm scores as well. Our clinical validation study consisted of over 600 samples from 4 independent cohorts. Both retrospective and prospective specimens were accessed. The MSDA algorithm configuration is a stacked classifier logistic regression model. 18 biomarkers are used to determine 4 pathway scores, as represented in the Venn diagram in the middle of this slide. The biomarkers and pathway scores are then used to determine the overall disease activity score. The output of the algorithm was strongly associated with 3 endpoints that are currently used to manage MS patients: active lesions on an MRI reflecting acute inflammation, new or enlarging lesions on an MRI reflecting activity that has occurred between visits or scans, and active stable status, which was a composite endpoint of any clinical or radiographic evidence of disease activity. The multivariate algorithm also significantly outperformed individual biomarkers on the panel, including neurofilament light. We developed a scoring process with 1 to 10 with thresholds for low, moderate, and high levels established that reflects both the likelihood and the severity of disease activity. Strong odds ratios were determined that are associated with the radiographic risk of disease activity. To summarize those, if a patient has a moderate or high score, they're 4.5 times more likely to have 1 or more lesions than a patient with a low score. And a patient with a high score is 21 times more likely to have 2 or more lesions than a patient with a low or a moderate score. For each of our 3 dimensions, comprehensive and easy to interpret reports are delivered to the clinician. This is an example of the MSDA test report. This is how results are summarized and communicated to the clinician. Key demographic info that is used to adjust the algorithm, the patient's age and sex, as well as information to help contextualize the result. The patient's disease duration, their current therapy is included in the top section. The overall disease activity score and level, high, moderate, or low, and an interpretation that relies on shifts and changes from prior results are captured on the first page. Longitudinal results are presented for each of the 5 scores. Individual protein concentrations are reported as well for each of the 18 biomarkers on the second page, with percentiles relative to the MS population that were established in our lab. This slide represents the MS patient journey that was developed and refined with input from clinicians who are using the test in practice, and displays how MSDA results can provide actionable insights. There are 6 use cases represented on the diagram, from baselining a patient, evaluating therapy response when starting or switching a disease-modifying therapy, confirming an MS relapse versus pseudo-relapse, and for longitudinal clinical management. Actionability is based off the decision points represented above each of the use cases and describes decisions that clinicians can make to optimize therapeutic interventions for the patient. Following up on the patient journey, here's an example of real-world data that was presented along with one of our partnering clinics, the University of Southern Alabama, Dr. William Kilgo. Both population-level insights and individual case studies were highlighted earlier this year at the ACTRIMS conference. Key takeaways from this are that the research was focused on underserved populations. 43% of the cohort is African American, which is a much higher ratio than what was noted in our clinical validation study as a limitation. We observed concordance between the MSDA results and clinical and radiographic evidence. 12 out of 90 patients that had been tested had high MSDA scores. All 12 had evidence of recent disease activity. Notably, 2 patients with high scores were observed that preceded the clinical radiographic evidence, hinting towards the predictive capability of the test. 3 case studies were highlighted of high MSDA scores that reflected different patient scenarios. The first is a high score that preceded a relapse. The second is a high score for a minimally symptomatic patient. And the third is a high score for a patient with highly active disease. In each case, the high score observed on the MSDA test is corroborated by the MRI obtained for the patient within close proximity to the blood draw. Pharma has also utilized the MSDA test to advance drug development efforts. This slide summarizes the first results from a clinical trial using MSDA that we co-presented at the ACTRIMS conference in the fall of 2023. This is post-hoc analysis of MSDA test results in a pivotal phase 3 study for ublituximab. That's a high-efficacy anti-CD20 monoclonal antibody that was developed and launched by TG Therapeutics. We analyzed 21 subjects from the ublituximab active treatment arm at up to 4 time points through 96 weeks of treatment. We observed very compelling treatment response trajectories. All patients were in the low MSDA category by their last time point measured. Additionally, replication of MSDA results was demonstrated for each of the validation endpoints that I mentioned earlier. Expanded results from this study, including additional subjects and a comparison to biomarker trajectories observed in the comparator arm of the trial, which were on a lower efficacy DMT, are being presented this month at the ECTRIMS conference. In addition to the measurement of disease activity in the clinic, our assay panel is also utilized by Octave and investigators to research individual biomarkers and multivariate models relative to endpoints of disease progression and disease severity. This slide is a summary of a study that was published earlier this year using individual biomarkers on the MSDA assay panel for analysis relative to clinical, radiographic, and neurofunctional endpoints of MS disease progression. This includes the EDSS scale, which is a disability scale used primarily in clinical trials, whole brain and regional atrophy, and neurofunctional assessments that reflect cognition, walking speed, and dexterity. As expected, GFAP, a structural protein uniquely found in astrocytes that has been widely reported on in neurodegenerative diseases, had the strongest associations with the disease progression endpoints we evaluated. Several of the other proteins on our panel were correlated with progression endpoints as well. We're currently expanding to larger protein panels offered by Olink, including Explore-HT and other immunoassay platforms in independent cohorts with endpoints that reflect disease progression to advance our development of a MS disease progression test. So in summary, Octave has leveraged Olink's cutting-edge proteomic platform to develop and validate a custom assay panel for MS. The MSDA test is available commercially and is used routinely in the clinic, as well as being leveraged by pharma for clinical studies. To further the development of our disease progression panel, we're casting the net widely again, leveraging Olink's deep scan capabilities. Our goal is to develop a new panel and algorithm with the ability to distinguish MS phenotypes from one another and predict the rate of progression. This would be yet another game changer for the management of MS patients. The bottom right of this slide has a volcano plot of results from the Explore 3K offering that were presented at the ECTRIMS conference last year. We demonstrated with our collaborators at University Hospital Basel that dozens of novel biomarkers beyond GFAP or neurofilament light are significantly associated with brain atrophy, both gray matter volume and white matter volume. These are key indicators of progression in MS patients. We're now leveraging Olink's Explore-HT offering that measures 5,400 biomarkers and currently analyzing data from an independent cohort that's focused on disease progression assessments in MS. We're also excited to report that we are now applying our biomarker test development approach to other neurodegenerative diseases, starting first with Parkinson's disease, and we're honored to have received support in the form of a grant from the Michael J. Fox Foundation. We recently ran our first Parkinson's deeply phenotyped longitudinal cohort utilizing Explore-HT. And in the coming months, we expect to be reporting initial results and sharing our progress with the community. I'd like to close out this presentation by acknowledging my colleagues at Octave. Developing and validating the MSDA test for commercial launch involved a large group with major efforts from our lab operations, data science, medical affairs, and several other teams. We're also very grateful to the crew at Olink for their partnership and collaboration over the years. Most importantly, I would like to thank the MS patients who contributed biospecimens and the research teams that provided the data from the numerous cohorts we have accessed for our studies.